coming. Our speaker today is Eduardo Colombo. He did his PhD in the Catholic University of Rio de Janeiro, Brazil, and at the supervision of Celia Anterior. He finished last March, and then he came here for a postdoc. Essentially, and myself. He also visited us last year for several months, so some of you already know him. So the topics of his PhD and the topics where he's working now is in the framework of theoretical biology, so nonlinear diffusion effects on population, on populations, metapopulation dynamics, effects of environmental fluctuations on population dynamics, and these kind of topics. And today he will talk about population survival in spatiotemporal environments.
Okay. Thank you for the introduction, Emilio. I was here last year. I was very impressed with the institute, and I'm very happy to be back here. And thank you, Emilio and Cristobal, for the opportunity to be here today and talk about population survival. So I took care of adding the new excellence prize here. So congratulations to IFISC for that. So the talk today will be regarding population survival, considering the spatial heterogeneity of the environment. I will start with simple problems, considering just an habitat with just a circular domain or just a simple domain in space. And we are considering several types of dynamics and several types of habitat considerations, such as seasonality of the habitat and so on. At the end, I will talk a little bit of more complicated problems, which now is which is known as metapopulation, where you don't have just one single domain, but you have several domains which are coupled and create some sort of network. So, okay. So just to give an introduction about the living beings, I will show you some pictures. I hope that works. Okay, so living beings have two, let's say, remarkable capacities. One is the self-propelling. They are self-propeller particles. They have internal structure that allow them to move in space. So this is, in the case of bacteria, this is possible because they have this kind of motors where they have the flagella. They can rotate the flagella and move around space. So this is possible because living beings have this sophisticated internal structure. Okay, so. And another, perhaps more remarkable property is self-reproduction. Living beings can reproduce themselves. This is a picture of a bacteria that is reproducing and is now left and can live by its own. So these two remarkable properties that I'm going to focus on my modeling. Of course, this also happens in large scales, not only for bacteria. You have birds, you have insects and mammals. You can study its mobility, you can study reproduction. There's some nest here, and there's many interesting things regarding this type of study. And I'm going to talk about the environment. Living beings are intrinsically coupled to the environment. You cannot think about a living being without the environment because living beings are in constant exchange of energy, consuming resources. So this coupling to the environment is very important. So, and the environment have also some remarkable features. One of them is heterogeneity. So here are some bushes. Bushes are here spaced by stones. And the bushes, if you note, there are different colors. So this is pointing out the difference of maybe water, maybe light emission here. So this is you can find different kind of habitats here, and here. And this heterogeneity is very present everywhere. Even just for a grass, a plane of grass, maybe a grass for a cow is homogeneous, but if you imagine an insect here, you have some holes. So this is also heterogeneous. And these are some tree distributions. So there are two species here. You can see some are more clustered, and this one's a little bit more sparse, but still more, there is correlation between them. So trees are not homogeneously spaced around. There are correlation between them. And here, perhaps more recently, it becomes important because human activity have changed the environment. And here you can see agricultural activity. So this creates these corridors where you have tree corridors, and it certainly affects the stability of the population in this environment. So this is some part of the changes that are caused by human activity. Many have call that we are now in a new geological area called Anthropocene, where the humans have crossed some sort of limit in their impact on the environment. So this is London. So you can identify here the Thames River. And the greener points here are greener region, let's say. So this creates a new type of habitat where individuals can live here and develop and reproduce and so on. So this is a new type of habitat that emerged because of the human activity. Here is deforestation in Brazil, which is a major problem in the Amazon forest. You can see that deforestation fragments the habitat. Now here is some fragmented habitat. But if you think about this region also, you see reduction of space. And of course, this is not a particular problem from the Amazon forest. You can see here, for instance, the loss of Greenland in the United States. So this look at the years, like. So you can see that when human population are much larger, the loss is very intense. Okay. Okay. And despite the interference in the spatial structure of the environment, humans interfere directly in animal behavior. This is some large collaboration, recent one, that shows that animal mobility decreases with human footprint. Footprint is characterized as like presence of construction, presence of human population around, and they quantify this index, and you show that mobility decreases with this index. So this is interfering in animal behavior. It is certainly will affect population survival, evolutionary dynamics, and so on. Okay. Despite that, environment is not a constant thing. They are changing in time. So just to show you a very good example, this is the temperature. When you look at the temperature, you can identify two main features that you find probably on every environment condition. One is seasonality, you can see here. So this is, okay, let me locate you. This is Copacabana Beach in Rio. This is one of the corners of the beach called Posto 6. There are several postos spaced by kilometers. This is one of the extreme. We have some fishery activity here, and there is a post of meteorology there. So you have a recorded data for like 12 hours, and this is like a cut of the data. So you see the seasonality, and you see some noise. Noise is intuitive to think because to quantify the temperature in a single point in space, the number of factors that you depend on it is so huge that complexity takes place, and you see this noise. So in seasonality occurs, of course, because of cycles of the planets and so on. And one interesting thing that you can look here is if you take the power spectrum of this time series, you observe the peak of 1 year. There's a peak of semi-annual oscillations that I didn't know, but it's present in some places. And there is the tail. The tail is exponential. If you imagine like which is for me is a non-trivial signature here, but it's not part of the talk today. I'll probably assume that environmental informations are just a white noise, for simplicity. But this is a natural situations. But if you want to take control of the situations, you need to make experiments with that. So this is for me, a very interesting work by Kimberly. I think it's in Kansas University. This is some samples, what she called fractal landscapes. So she studied the dynamics of an insect here. So she created these samples according to degree of fragmentations and so on. It's very interesting and beautiful. This is a very, let's say, more controlled environment because you can do it in a lab. So you create like a one-dimensional channel, quasi one-dimensional channel, and you put homogeneous distribution of resources, let's say for bacteria. So the bacteria have homogeneous resources, can consume resources equally in space, but you induce UV radiation and varying in space and time. So you just put a field here, but you place a mask. So below the mask, the bacteria will find a shelter from the UV radiation, but outside the mask, there is let's say a harmful habitat where the bacteria will die, the DNA will get damaged. So this is one possible structure that you can make. If you introduce time variability, there are some examples also that you can move the mask around like with a linear velocity and study the persistence of the population. And also you can rotate this mask. There are 2 examples. Of course, now with nano technology and nano advances, you can create nano landscapes, nano habitats for these populations. This is also one good example. This is just one-dimensional habitat, one-dimensional chain with some habitats here, and you can modulate these channels, making bacteria pass more or less through these channels. This is more recent experiments. This is you can see this is more related to metapopulation. You see that there are some organisms here, and they are connected to these tubes. So you clearly see the network here. And in this case, they study the modularity of the network and the influence of the modularity in the population dynamics. This one is very interesting. They put like a LED landscape, and the LEDs interfere in the mobility. So in this case, the organisms were attracted to the light. So this is very interesting because you can just place any type of structure as you want. You can induce blinking in the lights, whatever you want. So it's very interesting setup. Okay, so let's be more concrete now, what I'm going to talk about today. So let's talk about some basic concepts. So we are dealing with such a complex system. I'm not going to introduce all the variables that are present, all the factors. I'm going to adopt some kind of minimal point of view about the system. This is some sculpture that I like very much from Brancusi. Just with simple movements, Brancusi are able to express the shape of a newborn here. So if you struggle a little bit, this is the mouth here, the hair. So this is the newborn. So there's also this exercise from Picasso. He starts with a very detailed and complicated bull and start going to a minimal with simple lines that still describe the bull, but with few movements. So that's the perspective that I like and I will adopt here. So what we'll do, we'll describe the evolution of the population density in space. Basically, we will take the space and make a grid. And inside those grids, we'll define a local dynamics, but the individuals can move around. And what we'll do is to consider elementary processes as movement, reproduction, death, and competition in a very general way. Of course, this dynamics is conditioned to the environment, and actually is the main feature, the main problem that I would like to tackle now. So this is the problem. We have a dynamics conditioned to the environment, and we will look for the total population size and see the behavior of the total population size in the long time limit. And we are just would like to know which type of environment will lead the population to extinction or will lead the population to survival. So this is some examples here. In some cases, the total population go to a steady positive value, but sometimes it decay exponentially to 0. So this is the main question. So we will propose some different dynamics, we will propose some different environments and ask for the condition for population survival. And as a general thing in the problem that I'm going to show you, I will suppose that the environment is separable in the sense that we have a spatial structure and a temporal thing that are not like mixed together. So this is the type of things that we are looking for. So you have like the environment time scale, we have the environment spatial scale, and there are some regions where the population were able to survive or go extinct, given the population dynamics. Okay. So let's stop. So what model I'm going to use? A very simple and perhaps very traditional. Many of you know it. So we have like reproduction of the population, we have death, and competition for resources. This gives rise to the Verhulst model, and there is some stochastic fluctuations here due to the demographic fluctuations inherent in the process here. So you can write this in a different way using the carrying capacity. So if the environment have infinite amount of resources, the carrying capacity is infinite, so you don't have the saturation term, and the population will grow exponentially forever, but environment is finite, so it constrains environment growth. So, yep, And another step that we'll make now is that this is the model that I just showed you. So it's a Verhulst model with a stochastic fluctuation which comes naturally from the process. But I'm going to introduce now environment fluctuations, assuming that the environment interferes in the reproduction rate. So for instance, you have temperature. You can easily imagine that temperature is influencing population like a bacteria growth. So this is a very intuitive assumption. So you just move like the growth rate will be a constant plus a noise. What I do now is to make the Sartonovich formulation actually. So you assume that this noise have a correlation, but the correlation time is very small. So when you do this, you arrive automatically in the Sartonovich interpretation. So you have two noises. One is internal, so you interpret it as ito, but the other one is external. So this is the notation that I use, and the Verhulst model here. Many people are calling this the canonical model because it includes all the main features: environment fluctuations, the demographic fluctuations, and the exponential growth plus the saturation of the environment. So it's a very important model in population dynamics. And if you think about the population isolated, so if you look here, no individuals can emerge from the vacuum. Okay.
Are there any consequences that someone who's not into network science should?
Sorry, I don't get you.
Are there any consequences of this internal or external that someone who's not into network science should know? Like you said, this is internal and this is external.
Yeah.
Does that have any consequences? What you should relate to? What you have to know from the top level?
Yeah, because this is a dynamical noise, so it's from the dynamics. It's just mimicking the stochasticity of the problem. This one is a external thing that so it's have some correlation. That's the difference. This one is, some people would say, non-anticipative or anticipative noise. I don't know. I don't know what you mean about network science. Maybe later you will see something. So, okay. So if you look here, the null state is absorbent. So no individuals can emerge from the vacuum. If there is no individuals, you cannot reproduce. The population stay in the null state, which is absorbent. So if you start a population with a certain density or a certain number of individuals, it will eventually reach the null state, and the population will remain there forever. And you can actually calculate this using like some standard techniques to the first passage time. So this is the main thing that I'd like you to remember. If you consider a single population, which there is no individual coming, that you have a finite time, and the population will eventually get extinct. So let's introduce now the spatial coupling. So we just model one cell of this. Now we introduce spatial coupling, and I'm going to introduce it in a non-local way, so individuals can move around and non-locally in space, not just in the first neighbors. So you have the outgoing flow and the incoming flow here. And in order to understand better this expression, you can expand this kernel that couples the space in a Taylor series. And you see that this is just a generalization of diffusion. We have diffusion term here and superior order terms here. And it's actually like a diffusion, because if you solve just the equation for the transport, you see that all moments goes linear in time, and the rate that they go linear in time is given by the moments of the coupling kernel, this thing here. Okay, but of course this assumes random diffusion, random spread of the individuals. But if you have a good region here, and individuals know about that, they will not go, they will go to this region. This is something that I'll discuss later. Okay, but for now, let's be satisfied with the fact to truncate the transport term, the dispersal term in the first order, and plug to the Verhulst model, so we arrive at this equation. So you have the diffusion of individuals and the Verhulst model. So this is the main process that are being considered here, and this equation are known to have a wave propagation. Okay, so this is the main part. This equation leads to an homogeneous state, so there is nothing interesting here. If A is positive, if the birth rate is superior to the death rate, So the population will survive and we reach a homogeneous steady state if you consider, for instance, periodic boundary conditions here. What I'm going to do is to place the habitat now. So you have boundary conditions, you have heterogeneity, and things will change. So let's start the simplest case possible. So let's consider the line. I'm not going to do in 2D. It's very similar, but it's simpler in 1D. So this is the domain. So this is where individuals can reproduce. Here, the bacteria or whatever can reproduce. When the individuals leave this region, the bacteria immediately die. It's a strong approximation, but for now, I hope you be satisfied with that. So here, bacteria will reproduce, but when they leave the domain, it will immediately die. So this is the boundary conditions here, and I'm neglecting noise and everything, so you just have reproduction and competition for resources. So this is a problem that arises in quantum mechanics, maybe electromagnetic courses that we take in undergraduate courses. So this is trivial. You can solve this by Fourier expansion. So you have the modes here, but the modes are, as in quantum mechanics, you need to define the modes as the size of the infinite well, right, in quantum mechanics. So this is the mode that can grow, and there is the mode which is the fastest one, and this is one that you use to find the critical size. So for a critical size here, the population can, above this critical size, the population can grow. Oh, sorry, here, population can grow, but below this critical size, population decay. The critical size value is this one. So if you look at here, the minimum condition here, you need to consider the I0 here, and you just place here, and you are able to find the critical value of L. So this was the first problem done maybe many decades ago, and this is establish some sort of problem for the critical habitat size. So this is the main problem, and just to mention that if you look here, this is just a question of time scales. So the critical habitat gives the time scale, the correct ratio of time scale in which the individuals that reproduce before leaving the habitat. If this time scale is okay, so you have the persistence of the population. The individuals need to reproduce before leaving the habitat and die. So if you may argue now that I'm doing like a macroscopic equation or mesoscopic equation for the dynamics, but if you work with individual level, working with the individual level simulations, you actually see that the value of the critical size predicted by this theory gives the correct, let's say, threshold for population persistence. Okay, so let's make some improvements in this problem. So this is the first one that I would like to show. This is some theory on active matter. There are some people working on active matter and propose this type of model that represent bacteria that runs and tumble, but they are interact. So you see this higher order term here. So there is appearance of this type of domains and so on. So you can ask yourself, what's the critical habitat size for this type of dynamics? So you consider this equation with these boundary conditions, and you see this type of solutions. It's actually a part of some results that we did last year with Emilio and Cristóbal. And we are trying to understand these patterns that emerge in these experimental situations here with bacteria. And just to show you like some preliminary results, you can calculate, like in this case it's very simple, and you go and you find that the critical habitat size depends on this interaction, and actually the critical habitat size grows with the higher order interaction term. And this is just a way to show that the deviation that experimentally was found by Perry in his experiment can be given by this type of interaction. Okay, I'm going to be very brief in this one. So this is a problem that I'd like to talk a little bit more. So this is nonlinear dynamics now, and there is some very interesting problems that will emerge from this equation. So I'm going to consider nonlinear diffusion. So diffusion now depends on the density. So if the density is high, maybe it will make the diffusion higher or lower depending on this exponent. And growth can also be cooperative or be competitive in the sense that density can promote or decrease growth. So it's a very general equation that we proposed last year. And this is some example that I show you. So if nu is bigger than 1, so this is the solid line, the diffusion coefficient grows with the density. So this is the case maybe with gas in porous media and so on. But in other cases, the diffusion coefficient can decrease with ρ, which is the case when, for instance, the collisions of the bacteria or any individuals that are clustered together will decrease because they are like packed together. So this creates some general diagram that I show here, which reproduce some type of behavior of some animals. So these 3 animals here are known like some insects. Sorry, I forgot the names. They are very complicated. So, but they are known like to be subdiffusion. So nu is bigger than 1 for them. These mosquito have nu smaller than 1, and this parasite have mu smaller than 1 in the sense because in this case of this parasite, the growth is higher when the density is low. Why? Because the female can lay eggs around, but when the density is higher, the female becomes smaller and the number of eggs is smaller also. And with larger mu, you have the problem that the growth goes to 0 when the population density goes to 0. So there is some kind of loss of cooperation in the population. When the density goes to 0, the individuals lose the capacity of reproducing, and this is a feature known as allele effect. So this is a type of diagram, and you can reproduce many types of dynamics with this type of equation. Actually, this is the most difficult part for us as a physicist because we need to run the literature and look for animals that have this type of features. But we found some of them. So what we did is we found some generalized laws, of course, because the growth is not just linear, it's nonlinear. So you have some universality for some cases where there appear power laws. And for this case, it is a very strange one, but this is more clear. And also what we find is a generalization of the shape of the steady states, which in other case was just a trigonometric function, but in this case is more complicated. So this seems okay. So we have a generalized growth law, we have a generalized shape of the profile of the population. Okay, this is fine, but doesn't tell much. And what we did to find the critical habitat size was to apply this phase space method. What you do is to take a trajectory and find the places where the trajectory goes to 0. So we just rewrite the equations as this and make some change of variable. And so this is what I mean. I just propagating the solution from the 0 derivative to some derivative v and coming to z, from z0 to z, which I'm going to set 0 here. And I'm going to find which point is that. So when you do this, you find this point, which would be like L star over 2. But I'm taking care of not calling it critical size right now, because it's not an instability analysis. It's just the point where the solution reached 0, which might be or not might be the critical size. And this is what actually we find. We find there is a very general stability condition that I'm going to discuss, just to show you that sometimes this is the critical habitat size, but sometimes this is not representing the critical habitat size. So the full diagram is this one. So this is the mu-nu diagram for below this curve when mu-nu is equal to 1. We have survival of the population every time. It doesn't depend on the habitat size. So it's a very striking feature here. The habitat can be any, and the population will survive. Above this curve, we have conditional survival. There is a critical value where above which the population survives, and below the population will go extinct. But there is this region where everything changed. Above this region, when linearity is very strong, we change the problem. The population survives when the habitat is small and dies when the habitat is large. So this is it's a new thing where we are able to generalize the problem, because every problem that you find, you see that there is a critical size for the habitat, but this critical size is always the lower bound for population survive and not the upper bound. Another different thing that we find is that with the nonlinearity, the critical size of the habitat becomes dependent on the initial population that you introduce. So if you are a biologist, you are trying to reintroduce some population in a given habitat, the size, the critical size that population survive depends on the initial population that you put inside. So let's see. Okay, this is actually now the topic of the title of the presentation, which now we have a spatial habitat, but now we have a temporal scale also. So this is a model that we were inspired by the bacterial experiment that I mentioned already. That's the experiment here. So this is the channel, we have the mask, and you have the UV radiation here. But we ask ourselves if the mask is not static, if the masks are blinking or changing in time, disappearing or appearing. So it's like we call like intermittent habitat, or if you want a blinking mask here. So how this changes the population survival. And the model that we use is just without the fluctuations. So we have the Verhulst model here, the logistic expression, but we are coupled to a multiplicative field, which is the UV radiation here. But this field will have this temporal behavior. So for a period lambda tau, the habitat will be inactive, and for a period 1 minus lambda tau, the habitat will be active. So, but we are interested in the time scale. So we are going to fix these things and just move with the time scale and see how this is changed. So in average, the habitat is active in the same proportion of time. Of course, this mimics some type of seasonality, if you want. Also, this mimics like regulations. If you imagine like a reserve, and a reserve that you allow fishing or hunting with seasonal behavior, it's important to know the time scale that you will allow and the portion of time that you will allow population to be hunted or fished in this case. So it's a very cartoon thing just to illustrate. Okay, so this is the result that state the problem for us. So we fixed the amount of time that the habitat is active, fix the habitat size, and just move the time scale. So for fast time scale, you see the total population going to 0 exponentially, so the log scale here. So you see the population going to 0. But when the time scale becomes, the habitat time scale becomes smaller, you see like the oscillations, and the population starts to grow. So this is the first result that we took and caught our attention, and we started studying it in more details. So just to show you, this is the average. So I averaged the growth between the cycles here. So the growth increased, the average growth increased with the time scale. So you have here the slow, the fast limit and the slow limit here. So what's happening? What's happening is that when the population, the habitat is inactive, you have just an homogeneous field which is dragging the population down. So it's a very simple problem. Everybody is going, the growth rate is negative, which is -5 here, which is just the reproduction minus the damage of the UV radiation. So there is no spatial dynamics taking place. But when the refuge becomes active, you start to see a stretch. So the population inside the habitat starts growing, and the other one is dying, so you see a stretch. And this stretch causes a non-trivial behavior, what you see here, a response in the growth, that if you wait long enough, the growth will reach this phi zero here, which is let's say the steady growth rate if you wait enough. But if you don't wait enough, the population will just reach smaller values. Okay, so let's solve the static case. What's different here? As I just solved the static case, right, for the linear problem. But here, we don't have boundary conditions. We have a more realistic situation. So here, the bacteria will not die immediately. The bacteria receives radiation, but the bacteria die in a certain time. So you have some tails in the distribution of the population, and inside the population grow, and you can see like some peak inside. So this problem was like, is very old, the static case, and this is the field that reproduced this heterogeneous habitat. So what's the strategy here? We cannot solve the eigenvalue for this problem, as in the other case that we took the cosines, and we are able to find the rate of growth for each mode. In this case, we cannot do this. So there is a very a trick to do, is that when you have the critical habitat size, the population reaches a steady state. When habitat's a little bit bigger, and the population will grow. When habitat's a little bit smaller, the population will decay. But exactly at the critical size, the population have a steady state. So that is the first assumption. And knowing that, and you are able to get the solutions outside and inside, and you make the assumption of continuity of the solution, and try to find the critical size. So that's what we are going to do now. So this is the equation for inside of the habitat. So the population is reproducing and just diffusion. I'm taking the stability near the null state. So there is no competition at this point. So you have the reproduction and diffusion. Outside, you have the death. Population is dying and also diffusing. So these are the two solutions, and we are going to assume the steady state. So we take this to 0. And the solution is inside are trigonometric, it's just a cosine, and outside is an exponential. So it's here with different, let's say, rates for each one. So we assume the continuity of the derivatives here, the same derivative, and the same value of the population density. We apply this, and we find the critical size in which there is the steady state. The steady state exists with this critical size. So for our explicit case, we have like the population, as I said, there's this stretching. So there is a population growing inside, population dying outside, and for our case, these values are these ones. So this is the solution, and this is what Perry applied in the experiment. And actually, he found some deviations that I tried to argue that might be caused due to some other type of interaction that are taking place. So, okay, this is the static case, and this is the value. Okay, so let's go to the dynamic and temporal cases. So this is the refuge is blinking now. You have this behavior. So for the fast limit is easy. You just average. The population will see the average of the habitat. So the population will not have time to respond to these fluctuations. You just average the fluctuations. So the growth inside is just the reproduction minus the fraction of the time that the UV radiation is inside the habitat. So this is the average inside, and you just substitute it here, and you find this. It's very like standard when you have like some fast dynamics taking place. So for the slow case, it's a little bit more tricky because you have the population growing. If you wait long enough, the population will grow with the phi 0 that I mentioned previously, and will die with this term, which is the same here, if you wish. But this growth rate we don't know. You cannot calculate it. So, but if you know it, you assume that at a particular L here, it will get 0. So at the critical size, there is no growth. And you can find using the inverse function of this growth rate here. So what we did was to propose an ansatz for this growth rate, which fits well the numerical data. We use like some heuristic arguments to find this form, which is actually very, very nice. Miss a little bit here. But it fits well the growth rate as a function of L. So for large habitats, you see the population growing faster. And for smaller habitats, you see the population growing slower. So when we plug this to this solution here, we find this. So the critical value of the habitat for the slow, very, very slow fluctuations are just the static one times proportional factor here. So this is the two results for the fast and slow limit, and it was actually all that we got analytically. So the numerical results are here. So this is the two solutions for the fast and slow habitats, and here is the proportion of time that the habitat is not active. So these are the two lines, and we just take here is this represents the first diagram that I show you. So there is the environment spatial scale and environment time scale, and the population survive in this region and goes extinct in this region. So if the environment is low, if the environment is, oh sorry, I think, yeah, I think it's wrong. If the environment is low and big, the population survive and goes extinct if the population is small and the habitat is small and fast. So this variability seems small here, right? But if you take the relative value, you see that this variability can induce a 30% chance. So if you are an ecologist, or if you care about the preservation of a given population and you neglect these fluctuations, you can miss by 30% the size of your reserve or whatever you are managing in a real scenario. So, okay, we finally arrived in the metapopulation problem. So this is some nice picture of the work of Hansky, which did his career on metapopulation. This is a place in Finland where you have like some patches, which are those locations where individuals can live. So in this case are butterflies. So butterflies find good regions to live here, 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 in every black spot. But the butterflies can travel around. So you create a network. So you have subpopulations coupled together. So this is the scenario. So this is what I'm going to study now. Now we have the full and complicated model. So we have like for every region in space, every patch, we have a condition which gives the quality of the habitat. We have the competition for resource, and we have the spread of the individuals and the both noises, the demographic one and the environmental one. So I'm going to, for simplicity, assume a binary landscape. So in some places, the conditions are bad, and in some places, the conditions are good. So this is just change from minus to negative to positive growth rate here. So this is the dark spots here are the good places, and the white ones are the bad places. So to model the landscape, I propose like this Levy dust structure, which is it's very simple. You can just make a random walk jump in space, and the random walk jump according to this power law, and the only parameter that you have to control the spatial structure is the x-point move here. So you make the random walk, and just with this move, you can achieve different structures. So for a small move, you have like a very spread landscape, which reach the homogeneous case when mu is equal to 0. But when mu start to increase, you have a cluster landscape. Actually, if you want, there is maybe some questions around it. You can define a fractal dimension for this structure, which is given by this. So it reaches the plane 2D when mu becomes large. So this is the characterization of the landscape. This is the probability of the distance distribution. So this is the mu equal to 0. You have the linear case, which is the homogeneous case. But when mu start to increase, you start to see some peaks in the short distance, which are a sign of these clusters here. And why this is interesting? Because the properties and the qualitative aspects of this landscape resemble many features as the trees that I mentioned in the start of my talk, and also some properties of these new habitats that emerged recently in cities. So like power laws and so on. Okay, so another feature that I'm going to consider now is the information. So individuals now don't just move randomly. I'm introducing a parameter here, delta, which if you wish, you can interpret as a diffusive parameter. If delta is 1, this term goes to 0, and you just have a a diffusion generalized term. So just diffusive and random. But if delta is 0, you have this term which gives the flow in the direction of the good regions. So this is the gradient of the landscape. So the flow will be preferred in the direction of the better regions. So this delta controls the amount of information used by the individuals to go to the bad regions or not. So this induces some changes in the, let's say, in the network of the population. So if you consider only the favorable patches are coupled, so delta is equal to 0, so 0 diffusivity if you want, it's just a network. So you see the network here. But if D is equal to 1, the connectivity is much higher because the patches are coupled to the bad ones. So inside these circles, yeah, the connectivity is bad, but the connectivity is higher. So despite that, this will play a role in the dynamics. Okay, I'm coupling exponentially with the distance. So patches far away are less coupled, and patches near each other have strong coupling. Okay, so for the mean field case, so neglecting noise, you have this equation. You have the growth, the competition, and the diffusion. And if you analyze the stability near the null state, you find this type of problem that was studied by Hanske and Novaskainen 20 years ago, more or less. And you need to have that the leading eigenvalues of this, the coupling matrix, the distance matrix, need to exceed some certain value. The problem is that you cannot calculate this eigenvalue very easily. So some people propose like approximations. And of course, there is theorems in mathematics that allow you to make this type of approximation, which is that the eigenvalue is constrained about the sum of the rows of the matrix. So this is more or less what I did, but I don't calculate the minimum and the max. I just calculate the average to give an idea about the stability of the population. So this is this is the, let's say, the clusterization degree of the network, of the landscape, and this clusterization needs to exceed some value. So the clusterization needs to be bigger than a given value. So this is what theory is telling us, and this is what the deterministic simulations give us. So you have like the landscape is stochastic, so you need to make a sample average. So you have a fraction of population that survived in the landscape. So this fraction leaves the 0 after some mu. So when the population actually gets more clustered, you have population surviving here. And this is the first figure that I show you. What shows that in this case, which is mu equal to 1.7, which is here, which you have like about half and a half going extinct or surviving. So this is what you have, some like population going extinct and some population going to a positive finite state. And here at this point, all of them are surviving, and at this point here, all of them are going extinct. So in order maybe you to believe that this is a critical value for this mu, I can show you this plot, which is not very convincing because it's hard to get the time scale, but we believe that this is a critical value of mu, and this is exponents 2 to leave the survival phase. Okay, this is the deterministic dynamics. So the stochastic case, as I told you previously, if you consider just one patch, the patch will go extinct. So the population will eventually die. So this is what I want you to remember. But when I couple them, so this is the coupled case, you have diffusion between them. The population is going to a stationary state, or if you want a quasi-stationary state, because maybe if some fluctuations can lead to extinction. But you see a very defined quasi-steady state here. So coupling is stabilizing fluctuations and making population persist in time. So this is what I show here from, let's say, from local to global. From the local case, when D goes to 0, every patch will be extinct, but as you plug the populations, you start to see the stabilization of the total population size. This is just a relative value of the total population size. So you see the stabilization and you see an increase. So I'm going to discuss this difference on the delta parameter, which regulates the diffusion or information-based dispersal in this next plot here. So what we find, what was like very interesting, was that the maximum population is not obtained when you have maximum information. So the maximum population is obtained when you have a mixing of random behavior and biased behavior of information. So you can see here for strong diffusion is very clear, but for all cases you can find it. So the population size is maximized in a very non-trivial point about the information that individuals use to spread around. Okay, that was all. I'm just going to add some final remarks. So just remember the temporal oscillations can interfere strongly in the habitat size. We found a general class of stability for the problem of the single habitat domain when the dynamics is nonlinear. And for the metapopulation, we saw that we need a high fractal dimension, or if you want a clustered enough landscape to have survival of the population. And the maximum population is achieved when you have a particular balance between randomness and information-based strategy. So just to finish, I just mention a little bit about ongoing projects. This is a project that we are doing now with Emilio and Cristobal and Ricardo that was here today, and which we're studying the evolution of the phenotype in the population as a function of animal mobility. We are also studying some fundamental aspects of pattern formation that we started last year. And some of the problems that I'm still studying today is a problem of pattern fragmentation, that with nonlinear diffusion, patterns can become fragmented. So in all cases that many of you may perhaps have studied, you see some patterns, but the analytical solutions give some smooth exponential that reaches, that do not go to zero strictly. But with nonlinear diffusion, this can go to zero, and you have like patterns with density and zero and patterns, and you have this null population density barrier here, which is important for propagation of contact process and so on. We are studying some generalization of this process. Also some superstatistics in metapopulation, and as a hobby maybe, heat conduction in nonlinear chains. If you imagine a generalized bath, like not Gaussian bath, if you reach like Poisson baths here, there are well-known problems when you have nonlinearities here. You can have change of the direction of the currency, maybe some controversial things that the heat goes to the hot reservoir than to the cold one. So this is a very interesting problem for me. And there's some references that I mentioned during the talk. And thank you for your time today.
Thank you. And questions or comments on the talk? I have one.
Okay.
Is that in several places, well, we have used mostly continuum equations.
Yeah.
At some moments, some particle-based simulations. And well, in one place you have said that they give the same.
Yeah.
But I am sure this is not always the case. For example, do you know effects of in which the discreteness of the particles affect some of those results?
Okay. Let me go back here. Okay. I can just press. Okay. What happens here is that when this is for the linear case. Yeah, linear growth. You have normal diffusion. Everything is very simple here. And these are two models. This is the Gillespie one, but this is a work that Vopiani did showing the role of the number of particles. And you can see that the critical value is a threshold for let's say, a change of the behavior. But of course, the extinction time here do not go to infinity. The extinction time just only go to infinity when the number of individuals are infinity. So There are some other works by Katja and other people in the United States that show that perhaps there are some other measures that are more appropriate. And when you use these measures, there is some shift on the critical value of the habitat, but the main contribution still be the deterministic one. Perhaps for nonlinear problems that I show, perhaps for no other type of interaction, this can change. But for this one, the deterministic habitat size is a good reference, let's say.
Questions?
You show a linear equation with two noises. And you said that one of them you interpret as ito and another one as asymptotic.
Yeah.
I don't understand it.
Okay. Okay. Where was much before, right? Okay, this one.
Yeah.
Okay. Because if you like investigate, for instance, noise-induced transition or pattern formation here, and you like perform averages on these noises, the tone noise will just vanish. But the asymptotic one is important will be relevant in those situations. So, and you have a modeling difference between them. There are different origins in them, and they need to be considered in a different way. If you wish, you can interpret them both as ito or both as a certain object, but you need to add the correction to make them be like place it correctly.
Those references are something related to that?
Yeah. Actually, this notation I steal from Awazawa in the theoretical biology. He used the different dots to make a distinction between the noises here.
More questions? If not, thank you again.